Hello and welcome to Cabruman Aquarium's Discovery Lecture Series.
I'm Dr. Julianne Passarelli, the Education and Collections Curator at Cabruman Aquarium. Cabruman Aquarium is owned and operated by the City of Los Angeles Department of Recreation and Parks, and we are extremely grateful for the city's support. I would also like to thank the Aquarium Director, Chrislyn McCarran, and the Programs Director, Jim DePompe, for their support. And a huge thank you to everyone who has joined us tonight. The aquarium remains closed due to COVID-19, but we are making plans to reopen. We look forward to sharing information soon about our exclusive members only welcome back event. Please check our website for reopening information. Earth Day is coming up, and we encourage you to help us celebrate planet Earth by entering this year's digital art poster showcase. All mediums are welcome, including drawings, paintings, sculptures, photographs, etc. This showcase is open to all ages, and entries are due by Monday, April 19th. Visit our website for submission details. Before we get started with the lecture, I would also like to thank and acknowledge the Friends of Cabruman Aquarium for their support. Although the aquarium is closed, the gift shop remains open, and the hours are Thursday through Sunday from noon to 6. We also have an online store, and you have the option to pick up or ship your purchase. Your purchases support the aquarium and will help us get through this tough time. You can find the link to our gift shop on the aquarium website homepage, cabrumaquarium.org. We plan to continue the lecture series this spring 2021 online. The upcoming lecture is on Friday, June 4th, and our speaker is Dr. David Ebert from the Pacific Shark Research Center at Moss Landing Marine Laboratories. The title of his talk is Searching for Lost Sharks. Dr. Ebert is a world-renowned shark biologist and has authored over 30 books, including the forthcoming Sharks of the World. I hope you can join us for this online lecture. And then after that, the next lecture will be on Friday, August 6th, and the speaker is still yet to be confirmed. The schedule and speakers for 2021 are posted on our website. There's a link on the home page under Newsflash called Discovery Lecture Series, and that will take you to the upcoming schedule. If you missed any past lectures, they are all recorded and are archived on our website. On the Discovery Lecture Series page, just scroll down to Lecture Archives 2014 to 2021, or you can go to the CMA YouTube page. If you're interested in the upcoming lectures, you'll need to RSVP to receive the webinar link, just like you did tonight. But again, for now, all upcoming lectures will be online. It's my pleasure and honor to introduce tonight's speaker, Dr. Dave Cummings. Dr. Cummings is a professor at Point Loma Nazarene University. He received his PhD in microbiology from the University of Idaho, where he studied heavy metal transformations by bacteria in sediments of a mining impacted lake in Idaho. He was a postdoctoral fellow at the Idaho National Laboratory and studied bacteria that could be beneficial for bioremediation in contaminated groundwater. His current research focuses on bacterial contaminants from urban stormwater and coastal wetlands. The title of tonight's talk is Just Say No to Drugs, the Spread of Antibiotic-Resistant Bacteria into Our Coastal Wetlands. Thank you and welcome to Dr. Dave Cummings.
Thanks, Julie. Hey, everybody. Thank you for being here. This is really great. I was thinking as I was setting up my notes for tonight's seminar that it's been over a year since I've been able to give a talk on my research, and I mean, go figure with COVID, right? And so it just felt really good to sit down and start piecing together some of the key links in the bigger story of the work that we do. And I say we because it's always more than one scientist, right? There's a whole team of us working on these things together. I am a microbiologist, as Julie said. My training is in microbial ecology, so looking at the role of microbes in ecosystems. And I've always at the same time had a real interest in human health. And so it's been really neat for me to be able to study wetland ecology at the intersection of microbiology and human health, all together. And so that arrow is pointing to that intersection where my research fits. And hopefully, as we talk about this tonight, you'll see just that. You'll see that it's an ecosystem approach to understanding microbiology that could potentially have pretty far-reaching ramifications on human health. So why don't we go ahead and jump in? I know you'll have questions along the way. Feel free to put those in the chat, as Julie said. And as we get to the end here, there'll be time for us to continue to discuss this and try to address any questions that you guys have. Again, I really appreciate the opportunity to be here, and I really appreciate all of you guys taking time on a Friday night when you could be out, at the disco or wherever you would be, roller skating, whatever you'd be doing on this crazy Friday night. Instead, you're here thinking about bacteria in wetlands. You're my kind of nerds. That's just awesome. So let's just make sure we're all on the same page, right? I know in a marine biology series, like the Discovery series is, bacteria isn't always at the top of everybody's list. So let's make sure we understand what we're talking about here. Hopefully everyone's familiar with the idea of a coastal wetland. Coastal wetland really is just a swath of land along the coast that receives water both from the ocean and from the watershed above it. And the key features are that it remains saturated almost all the time. You can see, like at low tide in this photo here, there are mud flats exposed, but it's only a matter of hours before there's a new inundation of ocean water. So it tends to be saturated, tends to be very low oxygen, believe it or not. Other than the first few millimeters of sediment, almost everything else is low oxygen. And then high salinity, obviously, because of the ocean influence. And so these coastal wetlands are really characterized and defined by a complex, rich plant community that can tolerate those unusual conditions. What's really cool about these environments, they provide a lot of interesting, what you might call ecological services to us. And it's all based on this feature of trapping nutrients, ultimately. If you ever look at a map of a watershed as it funnels down towards the coast, everything that's picked up in that watershed ends up being washed down into that one hot spot that we call our coastal wetland. And there's a lot of nutrients on the surface. Land has far more nutrients, just as a general principle. Land, soil, has far more nutrients than water. And so the soil, as it all gets washed down from the watershed above, brings tons of nutrients, especially carbon, nitrogen, and phosphorus, to these coastal environments. And because of the way they're situated right at the water, right at the ocean, they tend to trap and filter out those nutrients. And because of that, the nutrients are enriched. And therefore you end up with this really rich, lush wetland of plant community. And then built on the plant community, you have the animal community as well. Like these birds here, we have everything from egrets to clapper rails and everything in between. They're just an amazing place. If you haven't spent much time in a wetland, find a wetland that has trails that you can walk, and you'll see exactly what it is that I'm talking about. So we've got these really rich habitats on the coast that trap nutrients. And it turns out that the same structure that leads to nutrients being trapped in our wetlands also traps pollutants. And at first you might think, oh no, that's bad. But generally speaking, that's one of the ecological services that we think about with our wetlands. Whereas, along the California coast, it's commonly cited that we've only got roughly 10% of our coastal wetlands left. The rest have been developed in some way or another. And in fact, I'm going to show you an aerial view of San Diego shortly, and you'll see the development within the watersheds that we've got here. And the idea is that when we don't have these wetlands, the pollution that our urban areas are creating, that pollution has nothing to trap it, and it washes into the ocean. It impacts the marine habitats. Very large currents pick it up, run it all up and down our coastline. We have beach closures after heavy rains because we've lost these wetlands. If you live anywhere near a beach community, you've probably seen these beach closures, or at least on the news seen these beach closures that happen whenever there's a heavy winter rain along the coast in LA, Orange County, San Diego, etc. And it's because we don't have our wetlands left to trap all of those pollutants as they come down. So they trap these pollutants and become what we call a sink. A sink is a place for those pollutants to just accumulate in a somewhat safe place, in a safe way, and hopefully not spread further, okay? But what I'm gonna talk to you about tonight, I'm gonna challenge that a little bit. I want you to think about Imagine you've got a filter at home on, I don't know, your Brita filter, right? It can only handle so much. It can only trap so much before eventually it's actually starting to become a source for those pollutants as opposed to a sink. It's no longer trapping them, it's releasing what it's got because it's become essentially saturated with those pollutants. So the question that I really want us to be thinking about, we're not going to be able to answer it tonight, is whether our coastal wetlands that we hope are acting as a sink for these kinds of urban pollutants, if they could potentially be playing the role of a source as well and redistributing those pollutants in different ways. Now, as a microbiologist, I'm going to share with you some biological pollution. Pollution that comes in the form of live bacteria. And granted, in this mud you're seeing in this image here, there's tons of bacteria already, the majority of which couldn't hurt a fly. They can't infect the birds, they can't infect you and me. But when we get this urban runoff coming in, there's the potential to pick up microbes that are interacting with humans in some way and potentially pathogenic disease-causing microorganisms as pollutants, but then also their DNA. And to think of DNA as a pollutant is kind of an odd idea. So I'm hoping that by the time I'm done, you'll see how DNA itself poses its own threat, regardless of what bacteria are carrying that DNA around. And then we need to be thinking about the plausibility of some of those biological pollutants being redistributed. And especially in urban areas, they don't have to be redistributed very far to come back into contact with humans, maybe through the birds that we see here in this picture, or through insects or animal populations, whether it's marine animals or you know, when I'm out here at the Tijuana River Estuary, we'll see foxes and coyotes. We can see all kinds of critters and they don't stay there. So you guys might remember the old, there was an old commercial years ago in Las Vegas that said what happens in Vegas stays in Vegas, right? Well, I have no idea if that's true or not, and I don't want to find out. But what happens in the wetland probably doesn't stay in the wetland. And I want you to keep that in mind, okay? So if the wetland is Vegas, it might not stay in Vegas. The idea is that things that interact with our wetlands, there are so many organisms, there's also wind, there's water currents, there's so many routes for those pollutants, theoretically at least, to travel and move away that we need to really think about whether these wetlands could also be acting as a source of pollution for us. So that theme will come up throughout the evening here as we chat through some data together. There are two wetlands that I've been focusing a lot of my efforts on because they're an interesting comparison and they're local, which makes it a lot easier to do sampling work. If you orient yourself here in this inset, you can see the United States. Here we are in Southern California at the Mexican border. Here's an outline of the city of San Diego. And you can see the San Diego River watershed, much of which, probably most of which, is rural areas, still used by people, but rural. A significant chunk of the city of San Diego gets drained into this river, and then it hits the coastline around Mission Bay, Ocean Beach, if you're familiar with that area. And there's a wetland there called Formosa Slough that I want to talk to you guys about. Formosa Slough is a very small wetland. I'm going to show you an aerial image of it in just a minute. It's like 1/10 of a square kilometer. This is like a little pond. And as the name implies, is a slough. It's a dead end off of this very channelized, very structured, human structured wetland system. And the watershed that you see here is relatively small. It's a little over 1,000 square kilometers, and it tends to be pretty rural. And there are no known point sources of any sort of biological pollution throughout this watershed. Okay, so keep that in mind as we compare it to the Tijuana River watershed. Much larger watershed. We're talking over 4,000 square kilometers, mixed use, lots of agriculture, and then a couple big cities, right? You got Tecate right about here, I believe it is. The entire city of Tijuana, which is well over 1 million people, and struggling to keep a lot of its pollution under control. All of this drains into the Tijuana River. And then further, the city of Tijuana has an agreement with the United States to take 25 million gallons a day of its raw sewage and put it directly into the river. The river right about where this triangle is for the Tijuana River Estuary, right about there, it crosses over to the United States. And there's the South Bay International Wastewater Treatment Plant there. That treatment plant is capped out at 25 million gallons a day. And so it diverts the entire river flow during the dry season, The only thing flowing in the river is raw sewage. It diverts the entire flow into the wastewater treatment plant. It treats it, and then it pumps it a few miles off the coast here after it's been treated. But when rains hit, this watershed brings so much water through the river that by the time it's reaching the Tijuana River estuary, a typical rain is going to be in the hundreds of millions of gallons per day. And remember, the capacity for the treatment plant is only 25 million gallons per day. And I've actually, because I watch these meters, these monitors, I've seen it spike to over 1 billion gallons a day. And you can imagine at that level, pulling out 25 million gallons of what's now mixed stormwater all jumbled up with their daily sewage does nothing. And so the treatment plant is actually shut down during heavy rains, and all of that water, including all the 25 million gallons of wastewater, flows out into the wetland. And now we're asking this wetland to perform a massive ecological service to us and absorb all that biological pollution, and it's just too much. A lot of it washes into the coastline. It washes up to San Diego, depending on the prevailing winds and currents. It can flow down the Mexican coastline. We have to close a lot of beaches. You guys have seen this routine before, and then it takes usually 72 hours after rainfall has stopped for things to be considered safe again because of all that human wastewater input. So we've got two very different wetlands, very closely aligned with one another, right? Geographically very close to one another, two very different watersheds. And it makes for some interesting comparison, as you're going to see here shortly. Now, let me go back a step actually and show you some images real quick. I want to show you Formosa Slough first. I'm hoping you guys can see my my screen here. This is Formosa Slough right here in the center of the screen. Remember I said this is only a little over 1/10 of a square kilometer, and you can see it's all built up urban all the way around it. If you're familiar with San Diego, this is the Point Loma neighborhood, the Point Loma area. Now keep your eye on Formosa as I'm zooming out. You see where it is. Here's the concrete channel to contain the San Diego River once it hits the urban area. And so Formosa is literally just a little back channel, a little dead end that both marine water and river water flow into and mingle and flush back out again. So yes, it's picking up local runoff right in its immediate neighborhood, but then it's also picking up whatever storm water is encountered in the San Diego River watershed. Now let's back out of the watershed a little. This is an aerial image, and it shows you just how much concrete and asphalt we're talking about. And concrete and asphalt don't play well with rainwater, right? Nothing permeates those, and so the rainwater runs across those surfaces, picks up all the pollution that it possibly can pick up, and in the meantime, it's running over parks and yards and neighborhoods and you name it. And it's all ending up right there at our coast. Now the watershed itself, I don't have a visual to superimpose, runs up here to Mount Laguna and back down again. And so most of this on the right side is all really wild open land, lots of trails and what have you. But once you hit the city, we get into a very urban area with Formosa Slough. So let's zoom back in so you have a good visual of Formosa Slough. By the way, our campus is right about here, right next to Sunset Cliffs Natural Park. I can see it right out my window right now. So this is very close to the PLNU campus. Now the Tijuana River estuary is much larger. So that Formosa Slough is about 1/10 of a square kilometer in the wetland, a little over 1,000 square kilometers in the watershed. The Tijuana River estuary, the wetland is about 10 square kilometers. This is massive. And the watershed is over 4,000 square kilometers and includes, like I said, a little bit of Imperial Beach, California, but then also Tijuana and Tecate. So here is that entire wetland. So when we sample here, we're not just walking from one site to the next. We're in a Jeep trying to get across, or we're in deep boots trying to get across this thing because of how large it is. Now, one thing I want to point out is that right here is where the river crosses. There it is running right through in the bottom right side of your screen, running right through, channelized the city of Tijuana. And then it comes right over to the South Bay International Wastewater Treatment Plant. And this plant, we can even zoom in on the plant and you can see it. This plant has a diverter on the river, diverts 25 million gallons a day right here to the plant, treats it and spills it back out through a pipe under the ground all the way out into the ocean, about 4 miles. But the plant gets shut down and the diverter is pinned open when heavy rains come. And so that water hits the estuary right here and just starts fanning out across this massive wetland here through all these channels and ultimately into the ocean. And if we back out, you can see just how urbanized this is. We've got, it's nothing but concrete. You see everything in gray because it's concrete that you're seeing in the image. And it's not until you get out past that that you start to see finally some rural areas. So these are the two sites where I've done a lot of my research trying to understand the impact of stormwater, particularly stormwater that in this case is carrying biological contaminants in our wetlands. So let me go back to my slides again. Hopefully everybody's able to see the slides again. Let's go ahead and look at some data. Everybody ready to hear some data and see what's actually going on in these two wetlands when it comes to antibiotic resistance. Let's start with one class of drugs. They're called Fluoroquinolones. It's not important that you remember that necessarily. You probably recognize the name Cipro. Many of us have taken Cipro. It's used commonly for everything from UTIs to food poisoning. There's just a variety of uses for Cipro. It's one of the more commonly prescribed antibiotics that we have. Here's a little diagram of it for those of you that are organic chemistry wonks. Now this picture here, this illustration shows some of the more common ways that bacteria resist the antibiotics. So the antibiotics target something, generally a protein, and they interfere with that protein's function. Often it's an enzyme. And so the antibiotic, you take Cipro, it's targeting a special bacterial enzyme called DNA gyrase. And all you really need to know about DNA gyrase is that it's essential for bacteria if they want to grow and divide. So if one cell wants to undergo binary fission and become two, and the population wants to grow in order to cause an infection, for example, then it's got to have this DNA gyrase to be able to manage its DNA. The fluoroquinolones like Cipro go after that DNA gyrase and freeze it up so it can't work anymore, and the bacteria just come to a stop. They can't do anything. Now, bacteria found some tricky ways to get around it. Now, I'm not sure how familiar you guys are with basic genetics. You're all familiar with the idea of a gene. Everybody hears about genes. They say, oh, I got my long, lanky arms from my mom, right? I inherited that gene, or I got the blue eyes gene from my dad, or whatever. That's vertical gene transfer from parent to offspring. What's a gene? A gene is really just a portion of a chromosome, just a stretch of code, A's and T's and C's and G's, that really acts as a recipe for how to make a protein. That's the whole purpose of a gene, is it instructs the cell for how to make a protein. So each of these resistance mechanisms is based on some protein that can protect the bacteria from the antibiotic. So for example, some Cipro resistance genes encode an efflux pump. It's a little protein that wedges itself in the membrane of the bacteria, and anytime the drug crosses over, the efflux pump grabs it and pumps it back out again. It's sort of like a sump pump underneath your house, right? So that the inside concentration is low enough that the cell can still function and survive. So it just keeps effluxing the drug out. So we see fluoroquinolone efflux pumps as an example. Another example with Cipro and the fluoroquinolones are proteins. They come from a resistance gene, produces a resistance protein, and the protein grabs hold of the drug once it gets in and puts a little modifying group on there. A couple of carbons actually is what it adds to it. And so by modifying the carbon on the drug, here's our drug, and if it puts carbon in some wonky place, the drug's not going to be able to interact with the DNA gyrase anymore, and it won't be able to kill the bacteria. So every time the drug comes in, the enzyme grabs it, sticks this little functional group on it, and neutralizes the drug, so the drug doesn't work anymore. We call those drug inactivating enzymes. So we've got efflux pumps, inactivating enzymes. And then with Cipro, we often see target protection factors. So the target is DNA gyrase, right? That's what this molecule of Cipro is going after, DNA gyrase. A target protection factor is a protein that binds to the DNA gyrase. The gyrase is still allowed to function, but the Cipro can't bind to it anymore. The Cipro can't interfere with it in any way anymore. So a target protection factor. So 3 different ways that these sneaky bacteria can avoid our antibiotics. And so in my lab, we said, well, are any of those genes, can we find any of the DNA that's evidence of these resistance genes actually in our sediments? As a microbiologist, you can go after the bacteria themselves, but there are real limitations to culturing all the bacteria. So often we'll sift through the DNA and search for the information that we're after. So what I'm going to show you in this graph here, we went to Formosa Slough and Tijuana River Estuary. At each of them, we went to 6 different locations around the wetland. And at each of those 6 locations, we collected 4 separate samples of the mud. And then we used something called PCR. Maybe you've heard of it before. It's a special DNA technique that lets us detect whether a specific gene is there or not. So you can see over on the right-hand side of the screen, the different genes, and you don't have to have these memorized. And in fact, it doesn't matter too much, but each of these genes represent different versions of enzymes or prescribe different versions of enzymes that can block the antibiotic action, like we saw on the last slide. So here's Formosa Slough at 6 separate locations. We simply call them FAM1 through FAM6. And then 1 through 4 is telling you how many of the 4 replicates were positive for each of the different resistance genes. Now, this is in the dry season. This was taken in the summer, at least 6 weeks after the last time there was any rainfall. So we're looking at essentially background levels. And the first thing you notice is there are some resistance genes there. They all seem to be this KEP-A, which is a quinolone efflux pump. So this is that efflux pump that I showed you. And it appears to be there at a few of the sites, 4 out of the 6, and with varying frequency in the replicates that we studied. So there's some background level of at least some of these resistance genes, even in the dry season at Formosa Slough. Now, the next image I'm going to show you will be in the wet season. And this is about a day, about 24 hours after a storm had moved out, after a storm had left us. Do you see the difference? I'm going to let that sink in for a second. Now, all of a sudden, we're not just picking up that one resistance gene, KEP-A. We've got 3 different resistance genes showing up with different frequencies and a little bit broader distribution across the 6 sites. Clearly, the stormwater impacted the distribution, the diversity, and the frequency of these antibiotic resistance genes. Now, keep in mind, we're in Formosa Slough. We haven't even looked at Tijuana River Estuary yet. This is an urban wetland in San Diego where the entire watershed is on our side of the border. No known point sources of biological pollution here. No known point sources. And we're still seeing our stormwater delivering resistance genes to our wetlands. You see those? Okay. Now, the next panel that I'm going to show you is going to be the Tijuana River Estuary in the dry season. And I wish I could hear your voices as you look at this, because there's Tijuana River Estuary without any rainfall. And you can see the names of the 6 different sites. That's not really what matters. We have 6 different locations spread across that gigantic wetland. We're looking at all those different resistance genes, and you see that all the genes are represented somewhere. All of them that we look for are there somewhere. We see much more frequent positive results, and we see it more diverse because almost all, let's see, 1, 2, 3, 4, 5 of the genes are there. They're at all 6 sites, some at least 1, if not more, at all 6 sites. And we see a lot higher frequency in terms of the number of replicates that we were looking at that were positive for those resistance genes. We're going to come back to that slide in a minute, because that slide, I think, is really one of the more important takeaways of what we think might be happening here. But before we do that, we have to look at Tijuana River Estuary and the distribution and diversity and frequency of these different resistance genes after rainfall. So what you see here is that almost all of them are detected again in much higher frequency. In fact, they're maxed out 4 for 4 for many of them all across the board. So we've got an even broader distribution of them, more frequent, and a high diversity still. So what can we say about this? Number one, we can say that these urban wetlands are in fact a sink at the very least. They're receiving antibiotic resistance genes from the watershed, even when there's no known point source like we see on the left side of the screen here with Formosa Slough. But then when there's a point source that's part of the whole system in the form of, this case, sewage, all of a sudden our numbers go up, our diversity goes up, our frequency goes up. the pollution, the level of pollution of this biological contaminant seems to be much greater once we do that. Now, if we were all live, I would say, okay, do you have any questions? And I'd pause here, but we can't do that. So if you have questions, just nod your head. I can't see you anyway. Write your questions down, stick it on the chat, whatever you need to do, and we can come back to this at the very end if there's some things here that you want to tease apart, some specific questions that are popping in your mind. All right, let's see how we're doing on time. 7:36, great. Okay, well, the fluoroquinolones like Cipro aren't the only antibiotics out there. And in fact, the penicillins and cephalosporins, which we combine those and we call these beta-lactam drugs. These beta-lactam drugs represent well over half of all the antibiotics we use, right? So anything that ends in cillin is a penicillin. So that's your amoxicillin, your ampicillin, your standard penicillin, etc. Anything that starts with ceph is a cephalosporin. So that would be like cephalexin, better known as Keflex, and a variety of others. So the penicillins and the cephalosporins, two of the most important, if not the most important, antibiotics that we use around the world. Now, there are several different ways bacteria can become resistant to these penicillins and cephalosporins, but by far the #1 most common way that they do it is represented right here. It's an inactivating enzyme simply called a beta-lactamase, right? The drugs are called beta-lactams. The enzyme that breaks them apart is called a beta-lactamase. So there's a beta-lactamase gene somewhere inside the cell. It gets expressed. The protein, the beta-lactamase itself, sits on the surface of the cell. And anytime it runs into a penicillin or a cephalosporin, it cuts it. And I can show you, for those of you that love organic chemistry, it cuts this bond right here between the carbonyl carbon and the amino nitrogen. And by cutting that little box there, all of a sudden the drug is no longer effective at killing the bacteria. So these beta-lactamases are extremely important, because when one of these shows up, it shuts down a whole branch of options for us for treating an infection. So I'm going to show you a little less data this time, but we did another study where we were looking for a beta-lactamase gene called CTXM. And there are several classes and variations of CTXM, but here's the issue. CTXM genes knock out one gene, one enzyme, and it can wipe out all the penicillins and almost all of the cephalosporins. So half of our list, or more than half of our list of options for treating a patient's infection, off the table. That's really bad news. You ask any doctor, they'll tell you that really handcuffs their ability to treat their patients well. And this CTXM variety of the beta-lactamase has just been taking off globally over the last decade. And I mean taking off. It was originally found in Germany, one little rare gene that showed up in somebody's infection that wasn't responding well to treatment. And literally over the course of the last 10 years, we've seen it spread from Germany to virtually every continent, including the United States. So these CTXM genes are very important, very dangerous, very problematic. And we thought, well, why don't we go for something that hasn't been around a long time, right? Those quinolone Cipro type resistance genes have been around longer. As far as we know, they've been more distributed longer. So it makes sense we'd start to see them kind of archived in the sediments. But what about the CTXM gene? It seems to have just sort of emerged in Germany and then covered the planet over about a decade. And so again, using PCR, we collected sediments from the Tijuana River Estuary and Formosa Slough, both right after a rainfall and well into the dry season. But then we also went to the South Bay International Wastewater Treatment Plant, which we know that the contents of that treatment plant are what spill into the Tijuana River Estuary during the dry season. And then we also got permission to go into the Point Loma Wastewater Treatment Plant, which treats most of the sewage for the city of San Diego, which we thought would be a good representation of what could possibly be showing up at Formosa Slough. And what you see is that we got a positive response from sediments in the Tijuana River Estuary in the wet season. Not shocking, right? Dry season, nothing. So you remember the slides I showed you earlier where most of those other genes, the quinolone resistance genes, were still present even in the dry season. We're not seeing the CTXM gene there in the dry season, only after the rainfall. Didn't pick it up at all in Formosa Slough, dry or wet. So it doesn't appear to be washing out of our sort of routine watershed that doesn't have any point source of biological pollutants. But then it was, in fact, not surprisingly found in the sewage at both treatment plants. So in addition to the quinolone resistance genes, we've got good evidence that this very important and realistically, honestly, dangerous resistance gene category is in fact showing up in our wetlands on the coast here. So I know it sounds like you're like, oh, I wanted some good news on a Friday night. This is all bad news. It's not all bad news, but it's important that we know what we're up against, right? Now, some of you may be thinking, well, what does it matter if this DNA shows up, right? If it's in bacteria that aren't going to cause infections anyway, maybe it is because we don't know the hosts. In some cases, we can identify the host. In other cases, it's just purely the DNA that we're looking at. Why does it matter? Well, bacteria have this crazy capacity to share DNA with each other. We call that horizontal gene transfer. So vertical gene transfer is from parent to offspring. Horizontal gene transfer would be like between siblings or neighbors or friends. Imagine if somebody's sitting with you right now watching this webinar, imagine if they could reach out and put their hand on your shoulder and transfer some DNA to you and you could start using it right away. I mean, it'd be crazy, like superhero stuff, right? But it doesn't happen. It doesn't happen in you and me, but it's actually very common with bacteria. And there are several mechanisms for that, but the most common is what we call conjugation. Conjugation is when a bacterium, like this little guy here, it has its chromosome, and bacterial chromosomes are circular, and there's only one of them generally. Bacteria often carry little mini baby chromosomes called plasmids. And these plasmids are almost like little viruses in that they can infect the bacteria and then spread to other bacteria. But unlike a virus, they don't actually kill their host. Instead, they help their host. They bring something new. So it'd be like you put your hand on the shoulder of whoever's sitting with you, and not only do you transfer some DNA, you give them some superpowers in the process, because that's what this is. This is a Marvel movie right here, okay? So the plasmid itself has all the genes necessary for what we call conjugation. Conjugation is when the bacterium that's carrying the plasmid creates a long protein fiber called a pilus. It grabs onto a nearby bacterium and draws it in like a fishing rod. It just reels it in until they're connected to each other, like we see in the second illustration here. And when they're connected to each other, the plasmid also contains other genes called mating pair formation genes that open up a little pore, a little channel from the inside of one cell to the inside of the other cell. And then the plasmid ensures that a copy of itself gets made, transferred across that little mating pair pore, and then the pore is closed, the bacteria are released from each other, and now both of them have the plasmid, the donor and the recipient. Now, in addition to those genes that are necessary for the transfer, plasmids often carry genes for things like antibiotic resistance, like what we're seeing here, virulence, which means the ability to cause disease. So genes that encode things like toxins that can allow bacteria to be more pathogenic. And then even some catabolic genes, which means genes that give bacteria different options for what they can eat. So it's competitive. It doesn't really impact what we're talking about here, but certainly the virulence genes do, along with the antibiotic resistance genes. The types of genes we were just showing you in the last couple of slides, all those fluoroquinolone resistance genes and the CTXM type genes typically have always been found on plasmids. So we asked the question, all right, are these on plasmids? Are they in a position where it doesn't matter what bacteria is washing out of the watershed and delivering these genes, because the plasmid could actually induce its own transfer to other bacteria, and this information of resistance could actually spread? And so the next thing we did was I wish I had time to really get into the details because this is some interesting experimentation. We actually captured plasmids from some of the bacteria washing in without necessarily having to identify the bacteria or culture them. We took bacteria that washed in from the watershed and we removed them from the sediments, and then we mixed them with a recipient bacterial strain, a lab strain that we can control very carefully. And then we actually just set up conditions for them to mate. And what we saw was that, and what we continue to see, because we still do a lot of these mating experiments, is that many of our lab strains that we mix in with the bacteria in the sediments pick up plasmids through this conjugation. Now, once that plasmid is in our lab strain, we're in a position to be able to break it open, study the plasmid, sequence it, right? Get the entire order of A's and T's and C's and G's and start mapping out genes. We have lots of these plasmids we've been working on. I'm going to show you two that kind of illustrates some of what we're learning from these plasmids. Now, this plasmid called PTRE-131, it's over 47,000 nucleotides long. So I'm not going to show you A's and T's and C's and G's. That's kind of useless. So we draw it out as a circular map because the plasmid is a circle, circular piece of DNA. Everything that's in green is what we call backbone genes. These are the genes that are just the basic function of the plasmid to make sure it gets replicated, gets passed on from generation to generation, that it can make that pilus, that it can retract the pilus, that it can make a copy and send it through the mating pair pore that we talked about. All of those basic functions for plasmid survival are on these green genes. The gray genes represent what appear to be genes because we can see a start and a stop, but we don't recognize them. In none of our databases do we have anything similar to them. So there's still a lot of really interesting discovery work that needs to be done to characterize a lot of those. And my lab is working on some of those. Now here's where it starts to get interesting now. These little yellow genes here are what we call mobile genetic elements. Mobile genetic elements, you may be familiar with a more common term that's used called jumping genes. These are little pieces of DNA that can actually jump out of one molecule like a plasmid and into another molecule like another plasmid or a chromosome. And when they do, they grab DNA around them and they bring it with them. So what we tend to find on our plasmids are collections of these mobile genetic elements in yellow, jumbled up with a bunch of what's in red that we call accessory genes. And over time, accessory genes that are useless and provide no selective advantage are lost. And the ones that are selected for and sort of fine-tuned tend to provide an advantage to the new bacteria. So you remember I said that when these are like viruses, but instead of getting sick, you get help. You get some superpower. The superpower that a bacterium gets when it's infected with a plasmid are all these drug resistance genes. Now, all these little acronyms don't mean anything to you, but let me explain just a couple of them to you. So this gene titled AAC, that's one of those quinolone resistance genes that we saw in earlier that we detected actually in both Formosa Slough and in the Tijuana Estuary. And there's that same gene on a plasmid from Tijuana Estuary. This OXA1 gene isn't the same as the CTXM, but it's a similar category. It's a beta-lactamase, and so it goes after penicillins and cephalosporins. CAT is a chloramphenicol resistance gene. SOL1 is a sulfa drug resistance gene. QNRB, if you're paying attention to those specific genes we were looking at in the earliest slides, the QNRB gene is one of them that we found in Tijuana Estuary and Formosa Slough, and here it is in its native environment in a plasmid. Quacky delta, there's two of those and another SOL1. Quacky delta is really cool in a bad way. Quaternary ammonium compound, better known as Lysol. This is Lysol resistance, folks. We have bacteria that have obtained genes that make them resistant to Lysol. And they tend to be mixed in with all these antibiotic resistance genes as well. And then they're found on plasmids, which we just showed can actually spread from one bacterium to the next epidemically, like a virus. This is what we're most concerned with. Not so much what live bacteria come in, because if, let's say E. coli, let's say this washes down inside an E. coli. E. coli is not going to like the marine environment and the intense sunlight and what else? Yeah, those are probably the two big things, right? The saline environment and the intense sunlight. They're not going to like any of that. And the E. coli will actually die off in a couple of days. But it appears that it's not dying off before it gets a chance to conjugate and spread these plasmids to other bacteria. Maybe they're native bacteria that are just living in the wetlands, minding their own business. Now all of a sudden they're multi-drug resistant and they can share this with other bacteria. Or maybe they're bacteria from the watershed that can actually adapt to this harsh environment or new environment in the form of a wetland. So there's one example. I want to show you one more that illustrates a couple other interesting things. This one, PTRE, plasmid TRE 2011, much larger, 143,000 base pairs. This is a lot of data here. Again, you see our green, which is our backbone functions. You also notice some green scattered in the accessory region, and that's what happens when those jumping genes pick something up from one place and shove themselves randomly into a plasmid. They can break up and interrupt portions, natural portions of that plasmid, and that's what we're seeing here. You see lots of these yellow mobile genetic elements, and not surprisingly, we see a bunch of resistance genes. Now, most of the resistance genes are up here in the, what's that, like 10 o'clock, 9 o'clock, 10 o'clock range, if this was a clock. We see another Sol for sulfur resistance. We see another quaternary ammonium compound Lysol type resistance gene. This AAD gene provides resistance to erythromycin and related antibiotics there. TAM is another beta-lactamase that is effective against penicillins and cephalosporins. We see streptomycin resistance, another sulfur resistance, another erythromycin, azithromycin, Z-Pak type resistance there. Tetracycline resistance. There's an arsenic resistance gene. Who knows what selected for that at what point in life? And look, there's one of our CTXM genes. Remember, we detected those in the rainy season. And now here on this plasmid, it shows up as part of the plasmid. Now keep in mind, any bacteria that picks up the plasmid doesn't just get one resistance, does it? It gets the whole menu of resistances. So anything that causes the bacteria to pick it up and keep it, all of a sudden it didn't just get one superpower, it got 5 or 6 or 8 of them all in one shot. So multiple drug resistances all coded on one conjugative plasmid. The other thing that's unique about this that I want to point out is this whole section below the CTXM gene. Now, I'm not going to identify what's happening in every one of those red marked genes, but I want to tell you those are virulence genes. In other words, these are genes that help the bacteria to cause disease. So for example, the HLY-F can cause pores or holes in your red blood cells. So if this gets in the bloodstream, it can actually get into your red blood cells and put big giant holes in them and cause them to burst open. Why? Because all of these genes, SRR, IUC, SHI-F, CYT-ABD, etc., these are good at stripping iron off your hemoglobin. Are you nervous yet? We've got a gene that produces a protein that breaks apart your red blood cells, and it's on the exact same element, this plasmid, with a bunch of genes that are involved in stealing and stripping away the iron from your hemoglobin. Okay, it turns out that this combination from about the CTXM down is a relatively common combination in UTI pathogens, bacteria that cause urinary tract infections. So here what we've got is a plasmid that could convert a run-of-the-mill, simple, innocent, minding its own business kind of bacteria into a multi-drug resistant pathogen all in one shot. These are the kinds of things that we're trying to track, we're trying to get a handle on, because remember, what we really want to know is whether or not these wetlands that are acting as a sink for our biological pollutants, clearly they're accumulating, could they also act as a source and redistribute them back into the human community? So what I want to do here is I want to hit some major takeaway points, because we talked about a lot. And if you're new to this discipline, your head's spinning a little bit with all these new things. What are the major findings from the work that we've been doing so far? Well, wetlands in urban areas are indeed sinks. And they're sinks for antibiotic resistance, whether that's the resistant bacteria or the DNA that makes bacteria resistant. And that's with or without point sources, because we saw this happening. We saw this distribution into the wetlands, both on the Formosa Slough side and on the Tijuana River estuary side. One had a point source of biological pollution, the other did not. Second take-home point or major finding is that sewage, and this shouldn't shock anybody, is an important point source of antibiotic resistance. And there are plenty of studies in the treatment plants to show that, but there's very little showing what happens if and when that wastewater gets out and away from the treatment plant. Another major finding is that antibiotic resistance is brought to the wetlands with stormwater and can accumulate over time. Remember especially that one slide, upper right-hand corner, that showed you Tijuana River estuary dry season. No fresh water, no stormwater had run down and brought sewage for weeks and weeks at that point. And yet there were still very high levels of very diverse antibiotic resistance genes there, suggesting that over the decades in which sewage has been a problem running into this wetland, they are actually accumulating. They don't just wash away. It's not a one-time event. And as long as you stay out of the wetland during the rain, which you should do, that somehow that's magically going to protect us and there'll be no consequences. The fact is they are accumulating, and that's going to change things. Now, I didn't show you data on the actual levels of these resistance genes. We have really cool tricks for counting them. We can actually determine how many copies of the gene there are there. I'll give you a couple quick numbers, but I didn't want to really bore you too much with this. That QNRA gene, one of the Cipro resistance genes, was at its highest level, most concentrated, most copies of it in the Tijuana River estuary after rain, shouldn't surprise us. And it was as much as 1 in every 50 bacteria in the sediments. 1 in every 50 was carrying a copy of that QNRA gene at that point. And you might think, oh, 1 in 50, that's not so bad, right? That's 2%. If you get a grade of 98 on a test, it's all good, right? When you're talking about hundreds, tens or hundreds of millions of bacteria per gram, and then just look at an image like this here and think about how many tons you've got, it's just absolutely mind-blowing to think about how many copies of these genes there are and what a significant proportion of the population is actually carrying them. The CTXM gene was highest in the South Bay wastewater treatment plant at about 1 in every 4,700 bacteria. And then in the Tijuana River estuary, remember there was a plus on that chart that said that, yeah, we detected that CTXM gene in Tijuana River estuary, but only after rainfall. Its abundance at that point was about 1 in every 23,000 bacteria, which is still well detectable and still quite high considering the millions and billions of bacteria in every small quantity that we're talking about. So these resistance genes aren't just a needle in a haystack. They are actually quite abundant relative to what's there. And then finally, the plasmids themselves contain multiple resistance and virulence genes, posing a little bundled threat, right? You know, Cox always wants you to bundle your phone and your TV and whatever else they want to give you, right? Unfortunately, these plasmids bundle virulence and lots of antibiotic resistance all into one shot to make it a lot easier for the bacteria to be transformed literally overnight. Okay, so let me just leave you with this question. Do we have an argument for why we need to look into whether these are just a sink and not also a source, or should we be looking at this as a source as well? I'd love to see if birds are redistributing them. There are a couple studies out of Europe that show that shorebirds, the bacteria in their gut resembles the bacteria in the sediments that they've been feeding on, including if there's been some wastewater released into those sediments. So it's very possible that these birds that are, you know, either year-round or seasonally feeding here are actually changing their gut bacteria to include all these resistance genes and then redistributing them elsewhere around the country as well. Lots and lots of work to do to figure out if this is not just a sink, but also a source. But I think we're on the right track. So I'm going to stop sharing at this point. I appreciate your attention. I'll tell you, I'm not a doom and gloom kind of guy. So I'm not going away sad or crying or thinking it's all not worth living, right? All is lost. But I am the kind of guy that thinks that we really need to know the truth. We really need to know what's going on, and it starts with us scientists. So thank you guys for your attention. I really appreciate you being here. Be happy to talk with you about any of this.
That was fantastic. Thank you. Oh, wow. I was writing down a ton of questions. There are some questions coming in too. I just learned so much. Thank you so much for that. There is one comment. Rosemary wrote, it's like a horror movie date night.
I'm sorry, I ruined your Friday night. So many ways
Yeah,
to.
there was a few times where I was, yeah, where you were like, I wish I could see your faces, and I was like, yeah, that's nuts.
Yeah.
Wow. Okay. So there are some questions. Let's see. We're going to try to get to all of them. Let's see. It's about 8 o'clock, so we're doing good. The first, I'm going to go in order, just kind of as they were coming in through your talk. So the first one really isn't about bacteria. Was what's the difference between a wetland and a marsh?
Hmm.
terminology?
Yeah, that's a great question. A wetland is a broader term. Basically, a wetland could be nothing but mud flats, right? A wetland is any land on the coast that's soaked all the time. And it doesn't have to have all the plant life necessarily. The marsh is usually a subcategory of the wetland, like maybe a subhabitat within it that has a lot of tall grasses. And those tall grasses, like you see in the images that I was showing, those are the marshy regions. So the Formosa Slough, I don't know how well you could tell from the overhead image, doesn't have much marsh to it. It's a lot of flat mud with a little bit of plants here and there, whereas Tijuana River Estuary is very marsh dominated because of the tall seagrasses or grasses that are there. I don't know if that helps.
Okay, and by the way, we could see all your images fine. It looked really
Oh, good.
Yeah. Okay, so there's a couple questions of people are asking, do we maybe need a bigger treatment plant at the border?
You know, it would help. It's just a bigger Band-Aid. You know, the issues are more issues around economy and poverty and those types of things. You know, when your government is really at its wits' end, just trying to keep people alive and safe in their community, it's really hard for any energy or resources to be put into something that seems a little less immediate or urgent, like treating the wastewater. So yeah, we could use a larger plant. We could use a better plant. Ideally, maybe we'd invest in more treatment plants in Tijuana and more infrastructure so that there are fewer and fewer people that are sort of off the grid and less waste that's just being exposed to the environment. A lot of solutions, and they're all very, very expensive. And unfortunately, money plays a role in a lot of these decisions.
Yeah. Okay. The next two questions are about your data, the graphs that you had up. So let me ask it, and maybe you want to share your screen again and see.
Okay.
So this is from someone anonymous. Why does Point South A in the Tijuana wetland show lower numbers in comparison to the other locations?
There we go. So South A showing less. Yeah, we don't know. That puzzled us for a while. And honestly, it would probably take a wetland hydrologist on the team to be able to figure that one out. It may be that that region, for some reason, is out of reach from the river flow and only gets more ocean inundation. It wasn't entirely clear to us either, but that was consistent actually. Whenever we went to South A, we didn't find nearly as much as we did everywhere else in the wetland. And I really think there's a hydrology, a water flow and distribution piece that we don't understand, and I don't have a hydrologist on my team. And I think that's what it would take to answer a question like that. It's a great question. I wish I knew where to go with it.
Okay, and then the same set of graphs. Dawn asks, FAM dry area 6 showed bacteria. FAM wet area 6 showed no bacteria. Just wondering why this occurred.
Yeah, another odd thing there. We wondered about that too. Like you could pick up this Kep-A gene here in the dry season, but as soon as the rains came, we didn't get anything. What I suspect, and I can't prove this, is that this gene here was in the native bacteria on the surface, and that the actual scouring of the water flowing through actually replaced it with a bunch of bacteria that weren't carrying it temporarily. And I bet if we went back, you know, maybe weeks later, we'd probably find a spike back in it again at that location. There's no way to know for sure, but you can see, hopefully right in front of you, why we replicate, why we take lots of different samples from different locations. Imagine if all I'd done was taken this one sample here from FAM6 after the rain, said, oh, I guess rain doesn't impact it, yay, right? No, we do lots of replicates, lots of different sites, try to cover all that heterogeneity. Great question.
Yeah.
You guys are paying attention.
They were absolutely, these were popping in right as you were showing these graphs. So
nice.
Yeah. Okay. Here's another one from Dawn. What is the major source of the bacterial pollution? For example, is it from human excrement or drug manufacturing processes or birds or animals? Like what, any ideas?
Yeah, so on the Tijuana River side, I can almost guarantee you it's such a strong hypothesis to say that it's the wastewater treatment plant, and therefore it would be, and that's all human excrement. So it's poop and pee, right? And that's why we found that one plasmid, the second one I showed you, that had a whole collection of genes responsible or commonly seen in urinary tract infections. Makes perfect sense, actually. On the San Diego side, on the Formosa Slough side, there probably aren't any point sources. It's probably much more diffuse. Everything from pet waste on the surfaces of lawns to gardens and manure that's used in the gardens to horses and small ranching operations, probably a variety of things like that. One of the options that you said as you read Dawn's question was drug manufacturing facilities. And even if those are spilling over, we don't actually find detectable levels of any of the drugs themselves in these wetlands. So it's not likely that the drugs themselves are here selecting for these bacteria, but the bacteria are generated up in the watershed somewhere, probably in our gut largely, and in the guts of other animals like birds and pets, and then washing in and concentrating in this one region right here.
Mhm. Okay, here's a question from Moss. Has HGT been verified for our ARGs in wastewater treatment plants? I understand that it has been confirmed in the lab via plasmids.
Yeah, there's evidence in wastewater treatment plants that it happens. There's also evidence that it happens in the human gut. So if you consume something that has bacteria that contain a conjugative plasmid and allow it to pass through the gut, you can then do a study on who it passed it to, and clearly it's conjugating even while in the human gut. Conjugation doesn't take a lot. They don't need perfect conditions, and it only takes about 15 minutes once they contact another bacterium to transfer that plasmid over. So it really does happen quite easily and quite rapidly and quite frequently.
Yeah, you just answered my question. So how long does it take? About 15 minutes?
About 15 minutes is what it takes once the donor and the recipient contact each other.
And how far apart can they be?
About one cell length. So they have to be pretty close.
Yeah.
But when you're talking about billions of cells per gram, they're all piled on top of each other anyway.
Wow.
They're living in very crowded conditions. These aren't the suburbs, these are the cities.
Yeah. Okay, Moss has another question.
Okay.
So the source of resistance genes found in the river and slough is overflow from the SBI wastewater treatment plant, or perhaps a broken conveyance system, correct?
Absolutely.
And the increase of stormwater flow just increases the probability of the sewage overflow at the wastewater treatment plant.
Absolutely. Yeah, as soon as it starts raining and any water starts to pick up in the river itself, it just overwhelms the treatment plant. And for safety reasons, they actually have to shut it down. And so everything at that point just goes into the wetland.
Okay, another anonymous person, is there food that you can avoid because you know these things come from eating wetland life or exposed to the bacteria? I think maybe thinking about fish.
Yeah, it's a great question. I'm not aware of any studies that have shown sort of a bioaccumulation of resistant bacteria or resistance genes. So there's nothing on my list that I'm avoiding. And I don't avoid going to these wetlands. I'm not going to go mucking around in them unless I'm wearing the right gear, gloves and waders and that kind of thing. But I like going for a run or riding bikes down at Tijuana River. These are places that are actually very safe to be around. It's just that they are very likely to be a long-term source of some of these problems that can just keep coming back into the human community.
Yeah. Okay, John has a question.
John.
Do you think transformation is an important mechanism for horizontal gene transfer in addition to conjugation?
Yeah. The general consensus among horizontal gene transfer scientists is that in nature, transformation is probably the least important of the three major mechanisms. The three being conjugation, like we just saw here. There's another one called transduction, where viruses actually shuttle the DNA between bacteria. That actually is very important in marine environments, and it's something I'd love to study in these wetlands. And then transformation is really popular in the lab, and we use it all the time in the laboratory once it was discovered to shove DNA into bacteria for various molecular biology techniques. But in nature, the general consensus is that it doesn't appear to be nearly as important in terms of the frequency of it happening as the first two, as conjugation and then followed by transduction after that.
Okay, another anonymous question. Are there certain environmental conditions in the wetland areas that facilitate the transfer mating of plasmids between bacteria?
What a great question. I'd say there's two conditions that make it somewhat favorable, at least. One being that you have the introduction of a bunch of donor bacteria, those carrying plasmids, from the wastewater flowing down the river. And then you've got a bunch of these recipients just sitting around waiting. And the other being that most of the bacteria in wetland sediments are living in biofilms, where they're piled on top of each other and stacked together in gooey, sticky layers on the surface of the sediment particles. And there's evidence that shows that that lifestyle, being that close together in biofilms, can actually promote more conjugation. It just makes it that much easier to share genetic information. And so as opposed to, say, just the water column, where biofilms are very limited in the water column, in the sediments, we've got a lot of biofilms. That's the predominant lifestyle of the bacteria. And that lifestyle has been shown to be conducive to conjugation and horizontal gene transfer.
Okay, there's a few more. Edith is got a comment or a question about if wetland birds are the ones distributing these bacteria. So what can be done about it? Bird protection takes priority over human health in wetlands.
Yeah, that's a great question. So my hunch is that birds are probably the biggest redistributor. I think insects might be an important one too. That study hasn't been done, but there are some studies, like I said, in Europe, starting to show that the birds are good candidates for relocating a lot of this, because when the birds feed on, horned snails or crabs or whatever, they consume sediment with it. And that sediment, as we can see in this graph here, contains all these bacteria with these drug resistance genes. And if they're on plasmids like we think they are, they can transfer into the normal gut bacteria, and they can become a permanent part of the bird's gut flora. And then it cruises around the world pooping on everybody's cars. And that becomes part of the dust that we breathe and the things that we contact indirectly. So I do think birds are probably very likely redistributors. What can we do about it is a tough question. I think that's where we got to draw in, conservation biologists and wildlife biologists to come up with strategies, because you can't block the birds from using these critical habitats, absolutely critical habitats. I think it goes back to the fundamentals of stopping the problem in the first place, so that we're not feeding the birds a bunch of antibiotic resistance laden sediment. But that goes back to the original problems of how do we keep our wastewater out of these fragile natural environments.
Yeah. Okay, another anonymous, along with sewage, much trash comes down through the Tijuana River. Have you looked at any of the microorganisms associated with the trash?
That's a great idea. We haven't. We've done cleanup days. Like, we're down there to collect sediments. We're like, you know what, let's just make a full day of it and just clean. We've hauled just bags and bags and bags of clothes and tires and everything else out of there. But we've never taken the time to actually swab and sample and extract DNA. That'd make an interesting study. So far, none of my funding agencies have funded us to do that specifically. They seem to be a little more interested in the sediment, but maybe there'd be an agency, I don't know, California Sea Grant or somebody like that, that would like to know what pollutants, what microbial pollutants the trash is dragging into our wetlands.
Yeah,
Cool.
does it attach to it somehow?
Yeah, whoever wrote that, anonymous, write a grant for that one. That's cool.
I know. I'll see if there was another question about plastics. Where did it go? I'll find it in a second. There was another one from Moss that I had the same question. Does horizontal gene transfer occur only between conspecifics, or can it occur between two different species of bacteria?
You know, one of my students asked me that same question today, actually. That's an important question. The more closely related the bacteria are, the easier it is to conjugate, without a doubt. So going from one E. coli to another E. coli is about as easy as it gets. Having said that, different plasmids have different what we call host ranges. So some have very narrow host ranges, and it's got to be a very close relative to be able to share the plasmid. Others have intermediate or really broad. There are even a few plasmids known that can cross over from gram-positives to gram-negatives and back, which are very, very phylogenetically divergent groups of bacteria. So it really depends on the plasmid, but our experience has definitely been that as a general rule, the more closely related the donor is to the recipient, the more likely you're going to transfer it, and the recipient will be able to express the genes that are on it. Sometimes we've seen the plasmid get transferred, but then the new host doesn't have the machinery to actually express the genes, and so nothing happens. That can happen too.
Okay, I found the microplastics questions from Christine. She says that was a great talk. Thank you. Do you think microplastics and the plastisphere sphere bacteria could play a major role in the redistribution of antibiotic-resistant genes as well? And do you think the antibiotic-resistant genes would persist during the transport to different environments?
Oh man. Yes, 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 and yes. That is a grant proposal waiting to be written right there. You know, we're looking for new and important avenues for the redistribution of these genes, and microplastics weren't even on my radar, but it makes so much sense. I keep thinking of things like wind and water and crustaceans and birds and fish, but microplastics, I mean, basically one pollutant redistributing this other important pollutant. I bet you anything that the answer is yes, because small surfaces, even when they're suspended in the water column, attract biofilms of bacteria, and those biofilms, like I said, are hotspots for conjugation and horizontal gene transfer to take place. So what would be really cool would be to be able to sample a bunch of those microplastics and examine them for their biofilms, but also for resistance genes. And then from there, you could just track the plumes of microplastics and where they travel to as a new distribution of these resistance genes. Very interesting idea. Very cool.
Yeah, there was a few about that.
That's great.
Alec has a question about plant life. Does the lack of plant life in the Formosa location contribute to its lower amounts of antibiotic-resistant genes, given that there isn't as much shade or marshy habitats where bacteria may thrive?
Yeah, it seems possible, Alec. Definitely seems possible. I don't have data to say yes or no, but that would make for a good hypothesis to go in and test somehow. You know, maybe you look in the marshier areas versus the mudflats and see if in the marshier areas you have more of these resistance genes accumulating than you do in the open mudflats. I don't think it'd be that tough of an experiment to do either, to figure that out. I like that idea. I like the question. I don't know for sure what the answer is.
Okay, we'll just do two more. Natalie has a question. Is there any research being done on more productive ways of treating waste?
Yeah, on the engineering side, there's not a lot of, there are microbiologists working on wastewater treatment because it is a microbial process. Not a lot of people are paying attention to DNA as a pollutant. I think that needs to change. The managers and researchers focused on wastewater treatment need to recognize that the DNA that's released poses a threat, whether it's through transformation, like I think it was Dawn that asked about, or once it's in bacteria, if it's through conjugation. And so there's engineering research going on to try to build a better machine, so to speak. And there's some microbiology research on fine tuning, but I'm not seeing anything dramatic or sort of paradigm breaking, groundbreaking to go in a whole new direction. One cool thing that many of you are probably familiar with is there are a lot of municipalities that actually use wetlands as part of their wastewater treatment, where they'll have a treatment plant and then the water, the effluent, especially if they're inland, unlike us, they're landlocked, they will run their effluent out that's been purified. And before it ever reaches either groundwater or a surface receptor like a stream or a lake, it moves through an engineered wetland, a designed human-made wetland, as sort of a polishing step because of that tremendous capacity that it has to trap all of these different types of pollutants. Like any filter, you could envision it getting clogged eventually. You know, I wonder if the Tijuana estuary is pretty clogged with this particular pollutant. And if it can really has much of a capacity to absorb more. But there are people using wetlands to polish them right off. And so, you know, these wetlands are powerful. They're really, really powerful things. We just need to know if what we're collecting is ever going to leave and come back to bite us, or if it's already leaving and coming back to bite us.
Let's end with an E. coli question.
Okay, I love E. coli.
Regarding conjugation, you said plasmids have the ability to adapt. Can E. coli, for example, adapt to heat and saline and survive in wetlands for a longer period of time?
I don't think I said that plasmids can adapt. I mean, there's obviously evolution of the plasmids. They change dramatically and very rapidly. The bacteria themselves, I mean, maybe what you're asking is, can the plasmids give the bacteria the ability to withstand things like more intense UV? And you know, there are some genes, UVRA, UVRD, I think of, for example, that are UV resistance genes. In fact, they were on one of the plasmids that I had up here, one of the images that I had. There are UV resistance genes that can increase the UV resistance of the bacterial host that's holding on to them by putting some proteins in the membrane that can actually absorb some of that high level radiation. Changing your entire habitat preference is a big, big step. And it's not something that a single gene or a single plasmid is likely to change. Those are major rearrangements in the chromosome. So if you look at the DNA of wetland adapted organisms and you compare that to something like E. coli, they're very, very different organisms. So I'm not convinced that a single plasmid is going to make E. coli be able to all of a sudden inhabit our marine wetlands. But boy, I've been proved wrong so many times. It's the joy of science. I'm usually wrong. So it's very possible that something like that could happen. I haven't seen it. And honestly, when we go into both Tijuana Estuary and Formosa Slough in the dry season, we can't find any E. coli. And we look, we can't ever find any. It's only during the rainfalls and immediately after that we can find it. So our data suggests that it is actually dying off after, you know, a couple of days when the rain stops.
Yeah.
But the DNA is staying. So clearly bacteria are dead, but hey, the DNA is sticking around.
Yeah. Yeah. Okay. I just want to read some of these comments to you so you can see them. Alex says, thank you. Adam says, that was amazing. Thank you so much. Cassandra says, I am in shock. Diana says, thank you for the webinar. And then Rosemary says, I'm taking microbiology, genetics, and ecology at the moment. Fascinating talk. Thank you.
Great combination.
Let's see. Wow, that was very interesting from Natalie. And there's one, I guess let's just end with this one. Tammy says she wants to know, can we do anything to make a difference?
I hope so. Like I said, I'm always full of hope. I don't see this as doom and gloom and the end of the world, but it's something we need to pay attention to and we need to be working on. And I still believe we've got to get back to the fundamental issues of, you know, this has been since the beginning of human settlements, getting our wastewater away from us. And what seems to be happening here is that our wastewater is impacting these coastal habitats that we may not be isolated from. And so if we can do a better job of getting rid of that waste, you know, whether it's in drying fields or dilution or better treatment or whatever, better containment, I really think ultimately that's where the best solutions are going to come from, not, you know, trying to keep the birds from flying away or treating all of our birds with antibiotics or whatever, right? I think it's going to come at the very, very source of the problem, the root of the problem.
Oh, Okay, well, we're going to end it there. I'd like to thank you again, Dave. Thanks again to Dr. Dave Cummings. And thanks again to Friends of Cabruman Aquarium and the City of Los Angeles for their support. We apologize if we couldn't get to all the questions, but we're starting to run out of time. And again, our upcoming lecture is on Friday, June 4th. It will be online. It's Dr. David Ebert from the Pacific Shark Research Center at Moss Landing Marine Laboratories. And the title of his talk is Searching for Lost Sharks. And then the next talk after that will be on August 6th. More information can be found on our website at cabruminaquarium.org. And a reminder, we have recorded this lecture and we will post it on our website soon. Thank you again for joining us. Stay safe. Hope to see you next time and good night.
Hi, guys. Thank you.